Thank you, Leslie, for this wonderful introduction. And I'm glad to be reminded that I'm fearless, even when I speak in front of huge audiences. It's an honor to be here, and I want to thank also the program committee for inviting me. I served in the program committee a couple of years ago, and I realized it's a lot of work, and it's very important work, so we should be thankful to them. Today, I'd like to tell you about our work in trying to understand the organization of neural activity across the brain. And before I start, I'd like to say that our work has been funded by a number of sources, including the Wellcome Trust, the European Research Council, NIH, UKRI, and recently the Simons Foundation, who's been amazing. And I would like to tell you that you can take pictures of anything you want in this talk, that I have no conflicts of interest, but I have something to disclose in that I have an unfair advantage over most people, which is, and the unfair advantage is that I share my lab with Kenneth Harris. And I've been sharing the lab with him for over 10 years now, and it's been great fun. And what you will hear today would not have been possible without him, and also without all the people in my lab. And I tried to have their names on every slide, but if I forgot anybody, I apologize. So when we prepare these talks, we're told to give a bit of a historical perspective, both on the history of neuroscience and also on our personal trajectory. And so I'm going to start from 100 years ago, which I think is only fair. About 100 years ago, two incredible papers came out, 3 years apart. In 1926, Adrian published this paper. He was the first person to record from a single neuron, and this is a neuron that tells you how stretched the muscle is. And he applied a load, and he saw that the number of spikes per second increased, and he realized that the neurons signal through action potentials, and it doesn't matter what the size of these action potentials is or their shape, it's always the same, but what matters is their frequency of firing. So this is the first plot that plots firing rate of a neuron versus something. And 3 years later, Hans Berger produced this paper in which he announced this incredible invention, the electroencephalogram, and this incredible discovery, which is that the brain is spontaneously active and generates its own rhythms. And he recorded from a dog where he had exposed the brain, and from his son Klaus, who I think was probably closing his eyes because this is a recording done over occipital cortex, and these are alpha waves, which Berger discovered and named. So these two discoveries have set the stage for two traditions in neuroscience that have stayed rather separate until recent years because they ask different questions. The Adrian tradition asks, how do neurons encode events? And these events could be visual stimuli, or body movements, or places during navigation, or values, and things like this. And the other tradition asks, how do whole neuronal populations reflect internal brain states, such as arousal, attention, resting, and sleep? Now, I was trained firmly in the first tradition, the Adrian tradition. In fact, Adrian trained Barlow, Barlow trained Blakemore, Blakemore trained Movshon, and Movshon trained me. I don't know if he's proud of that, but anyway, he did. And so when I opened my first lab and then my second lab in San Francisco, I started to move from recording from single neurons to recording from whole populations. And I would implant these 10 by 10 electrodes called Utah arrays in the visual cortex of a cat that was anesthetized. And we would record from about 100 neurons. And each of these neurons would have a selectivity for the orientation of a stimulus. You know this from textbooks. And so, but by recording from all these neurons at once, we were able to flip the question and ask not what does one neuron do when you change the stimulus, but rather what does an entire population do when you show one stimulus. So let me give you an example. We would, for example, show this stimulus here that is called the plaid. It's made of a grating like this and another faint grating like that. And we would study the population activity of these neurons, and which, you know, is it's very simple. Every neuron here appears in a different place on the x-axis, depending on what orientation it prefers. And when you show a stimulus that contains two orientations, you get two bumps, one at this orientation and one at this one, although this one is lower contrast, so the bump is smaller. Now, I was doing visual neuroscience until then, meaning I was studying how this activity would vary depending on how you change the visual stimulus. But there was one thing that was nagging me and the field in general, which was the fact that these responses are quite variable from trial to trial. You show a visual stimulus, you get a certain response. You show the same visual stimulus, you get a different response. And only on average, the responses would look like you see on this red curve. And this was thought to be because neurons were thought to be noisy, which drives me nuts, because in fact, if you inject a current in a neuron, you get a beautiful spike output that is always the same. But at the time, there was a thought that there was some noise generator, and so that a neuron, for example, this neuron here, this dot here, would have its own tuning or selectivity for visual stimuli, but then that selectivity was somehow corrupted by some noise. But the beauty of recording from a whole population at once is that you can see on a trial-by-trial basis what changes. And when you record, for example, 2 trials of the responses of this population, you see that the noise is not independent. Sorry, I say noise again. It's not noise, sorry. The variability, the something, is not independent. All of these neurons in this trial are firing more than all of these neurons, for example. And so I'm going to show you now probably the most complicated graph of the whole talk, and I apologize for doing it so early on, but the summary of this work is that we were able to replace the noisy neuron with multiplication and addition of signals that come from somewhere in the brain, and the rest of the talk is about telling you where and what, and that affect all of the neurons in visual cortex at the same time. So the neuron will have its own tuning or selectivity for visual stimuli, but also it will receive a number G and a number A that comes from the rest of the brain, and the number G will scale its responses multiplicatively, which you can already start seeing here, and the number A will scale the responses additively. If I've lost you now, too bad. No. If I've lost you now, here's a summary of what I said. Neurons in visual cortex not only encode visual signals, but they also encode non-visual signals that are shared among neurons, so this is not independent noise, and these signals provide addition and multiplication. So what are these non-visual signals about? We couldn't answer this in an anesthetized cat because there's no behavior, there's no nothing. There's some fluctuations that you don't control. But work from the Stryker laboratory and then from the McCormick lab, Carandini and Tolias, gave us the idea, actually they published this, that all these fluctuations are controlled by brain state and behavioral state. Now, to investigate this, we now are going to turn to mice and to awake mice. And we're going to use 2-photon imaging to image the activity of a population of about 12,000 neurons in the primary visual cortex of the mouse. So here, these twinkling dots are neurons that are firing. The mouse is looking at nothing, so these are not visual responses, okay? I'm not going to show you the activity of 12,000 neurons, but I will select a subset of 1,500 of them, and I will plot their activity as a function of time in what looks like one of the traditional raster plots. So black means firing a lot, white means not firing. And so here are neurons, here's time, and you can see that there's something going on. There's an alternation of activity. Sometimes these neurons down here fire, but then they shut up. And when these fires, these ones up here don't fire. And when these ones shut up, these ones fire. They alternate. And so you can capture the overall activity, not driven by visual stimuli, of this population by taking the first principal component of this matrix. Now, while we were doing this, which is this thing that goes up when these neurons fire and down when these neurons fire. So what is it? Why are these neurons going up and down like that? While we were doing this imaging, the mouse was free to run, so we could measure its running speed and its pupil dilation. Turns out the pupil dilation the two are very correlated. The pupil dilation is an even cleaner signal. And you can see that it's identical, essentially, to this first principal component of the population activity. Just to recap, the blue curve comes from recording from neurons, the red curves comes from taking a video of the eye. And so this thing that looked like spontaneous activity is extremely coordinated by the level of arousal. When the pupil is dilated, usually the mouse is running and it's certainly awake. When the pupil is constricted, the mouse is generally not running and it's probably drowsy. But now you may ask, what happens when you show a visual stimulus? How does this modulation by arousal interact with the visual responses of these neurons? After all, the day job of these neurons is to do vision, right? So now we're going to look at another population of neurons, I think about 1,800 neurons, while we're showing a visual stimulus of different orientations, and ask, what is the effect of running? And we're going to sort neurons by their preferred orientation. So here you see 1,800 neurons sorted by which orientation they prefer. Black is their firing rate, so black means firing more. You can see, okay, there's some orientation selectivity when the animal is not running, but when the animal is running, the firing rate goes up on average, and the visual responses seem stronger, but also there's this other effect. Some neurons elevate their mean firing rate very much, others not so much, right? And so to study that, we can look at the same neurons and resort their order according to how much they're firing during running versus how much they're firing during stationary. And so now you see that there are some neurons that really increase their firing rate a lot during running, regardless of the visual stimulus, and some neurons that maybe do the opposite. Now, for this aficionados in the room, I can say that this entire data set can be summarized by 3 curves. 3 curves for stationary, 3 curves for running. And that's done by taking principal components again. So the first principal component, the one that describes the activity the most, is not about vision, even though this is visual cortex. It's about whether the animal is stationary or running. And the second and third principal components instead are about, one is a sinusoid, the other is a cosinusoid, and they're about selectivity. And so you can recognize here, again, an additive effect, which doesn't care for what orientation you're showing. You just add spikes to a bunch of neurons or subtract them for some neurons. And there's a multiplicative effect. You make the visual responses stronger. So a summary up to here is that neurons in visual cortex encode non-visual signals related to brain state and behavior, and these provide addition and multiplication to the visual responses. So what circuits might provide this addition and multiplication? So an immediate suspect is inhibitory neurons. And the reason why I say it's an immediate suspect is that if you go interfere with inhibition, you find effects that are additive and multiplicative. This is something we showed in 2011 in my lab in San Francisco. We were recording from about 100 neurons, and we measured their orientation selectivity, which here, for simplicity, it's been rearranged so as if all the neurons preferred 0, okay? And this is orientation selectivity and direction selectivity. And then we applied gabazine, which is a GABA antagonist, and you can see what happened. The responses went up in both an additive fashion, so in response to all stimuli, but also in a multiplicative fashion, right? This peak here is bigger than this. Now, this is a crude experiment, very kind of 1980s in style. You're pouring some chemical over the cortex and you're seeing what happens. A year later, the lab of Massimo Scanziani, with whom I've collaborated many times, produced a fantastic study in which they asked a much more specific question in what specific neurons do, and they looked at the parvalbumin neurons, which are interneurons, which are the largest family of inhibitory neurons, and they asked, what happens if I make them fire less? What happens if I make them fire more using optogenetics? And when they made parvalbumin neurons fire less through inactivation, you find a very similar effect to the effect we had seen with gabazine. And when you make them fire more, you find a suppression of the activity. And again, these effects are both additive, subtractive, but also multiplicative, right? You can see that to go from this curve to this curve, you would need to subtract a number and divide by a number. Okay, so now we've moved from looking at GABA in general to looking at one specific cell class, the parvalbumin interneurons. But it turns out that different classes of interneurons react differently to behavioral state. And the Stryker laboratory, again, has been ahead of the curve on this. And so we need now a method to understand what the different interneurons do in reaction to changes in arousal. The problem is, number one, that there's a ton of interneurons. These are all the subtypes of interneurons present in superficial cortex in V1, okay? So you go from parvalbumin, somatostatin, LAMP5, VIP, SNCG, but when you go down to the types and the subtypes, you end up with tens of subtypes. So that's one challenge. The other challenge is that you can do two-photon imaging in vivo, like I showed you, and you can even distinguish excitatory from inhibitory neurons by making the inhibitory neurons red, for example. But you can't assign from two-photon imaging an identity to these specific ones. I mean, you could use a transgenic line that labels one class of interneurons, but you still wouldn't know who the others are. And so this has been a serious problem until extremely recently. And again, here is a place where my collaboration with Kenneth has been fantastic because he developed a whole side of the lab that I wouldn't even have gone 1 mile near, which is to create an in situ transcriptomics pipeline. So what that means is that you take, after you do two-photon imaging, you take the brain and you slice it up, and then you apply a system by which you find the expression of each RNA type. And when you find these clouds of RNA expression, you know that that's a neuron. And by the specific composition of that cloud, you can say which type of interneuron it was. So for example, we can say that this response of this neuron here was of a neuron, and now I honestly need my glasses, of type SST-TACHTR1D, which probably there's a few posters on at this meeting on this type of interneuron, okay? But then you can repeat this and look at the other neurons, and you know, all of these neurons are all the SST types, but you know exactly which ones they are. And you can assign now a class, or a subtype actually, to every single inhibitory neuron that we have imaged. Now this is very powerful, but the advantage of doing all this fancy stuff is there only if it leads you to simplify matters. If it leads you to complicate matters, it's better if you didn't do it at all, right? So it's ideal when, after this work is done, you can find some general law that allows you to not learn the names of all of these things. I have a real problem with remembering names, as you probably already figured out. And so we were happy to see that there is a general rule that governs what these neurons do during states of arousal and what their name is. And so first of all, let's peek here. These green neurons, which are parvalbumin mostly, seem to be more active when the animal is not running. Same with some of these purple ones, a few of these brown ones, whereas the blue ones and some of the green ones and some of the red ones seem to be more active when the animal is running, okay? And in fact, we can say that this period here is a period of wakefulness and arousal, and this period here is a period of particular non-arousal or drowsiness. It's actually a period in which the excitatory neurons, of which we have thousands, by the way, I've just squished them here, are showing an oscillatory activity. So we can now do two things. First is we can plot an index of how much each of these inhibitory neurons is active during the awake state versus the oscillatory state, and that comes from the imaging, versus one factor, which is the first principal component of their transcriptomic signature, which tells you about the expression of certain proteins versus others, okay? And comes entirely from the transcriptomics of the Allen Institute. And so we can now plot one against the other. And you see that here, the state modulation correlates strongly with this transcriptomic axis, which has to do with the expression of specific proteins. And this comes from a paper from the Allen Institute here, and this comes from our paper here. And they're in close agreement, which makes us now think that there is a general rule that simplifies this whole story. So let me now tell you this rule, and I'm going to drift into speculation as I tell you the rule. So this is the finding, which is that we can arrange these types and subtypes of inhibitory neurons across along an axis which is purely given by their transcriptomic expression. And I do realize I'm not explaining that, but it is in the paper. Okay, this comes straight from expression. And we find that that correlates strongly with whether those neurons are more active during oscillatory states or during alert states. But if you look in the literature, you can also see that these neurons have different levels of expression of acetylcholine receptors. So these blue neurons here and the purple ones tend to have more excitatory acetylcholine receptors, and these green ones and the SST-TAC and these PALVABs tend to have more inhibitory acetylcholine receptors. And again, looking in the literature, and again here the Allen Institute has been incredible, you can see whether there's differences in morphology. And you find that these neurons up here tend to send their axons towards superficial layers, whereas these interneurons down here tend to send their axons to deeper layers. And therefore, they hit pyramidal cells in different places. In the superficial layers, where it is thought that they receive mostly top-down inputs, and in the deeper layers, where it is thought that they receive mostly bottom-up inputs of the kind that drives them to spike in response to visual stimuli. So now, we're in the domain of speculation. I promise I will get back to data in a minute. Let's play with this for a moment, and let's imagine that right now you're extremely awake. So there's a ton of acetylcholine, okay? Ton of acetylcholine, you're incredibly alert, and all of these types of interneurons are firing a lot. That means they're shutting down the superficial layers, and therefore your layer 2-3 pyramidal neurons are getting mostly bottom-up inputs from your eyes, for example, indirectly. Now, let's imagine that you're super sleepy, or anyway, checked out. There's no acetylcholine, so your cortex is in an oscillatory state. Now, these types of interneurons will be active, and they will provide inhibition down here, and then you will get mostly top-down inputs. Okay? So I'm going to go back, awake, oscillatory, alert, oscillatory. No acetylcholine, oops. Acetylcholine. No acetylcholine, acetylcholine. Okay? I think so. I can't see you. I imagine that's okay. Anyway, this is speculation, but at least it hints at the fact that there might be a general rule that allows us to not learn the names of these tens and tens of inhibitory interneurons, but have some kind of functional understanding. Now, let me summarize up to here. We talked about inhibitory neurons. I told you that they are very good candidates for controlling addition and multiplication. And I showed you that they depend on brain state and behavior in a lawful manner that is controlled by one transcriptomic axis. So I have now a couple of questions. I told you about all these non-visual signals. It seems like they're a bad idea. Do they interfere with visual behavior? I mean, you have this beautiful visual cortex with orientation tuning, selectivity of all kinds. Why would you want to, first of all, I'm not going to be able to answer why, but then let's just say, let's pose a question that I can answer. The fact that there are these other signals, does it interfere with our ability or the brain's ability to do visual behavior? And the next question is, given that these signals are present in the visual cortex, might they also be present elsewhere in cortex? So to study this, we need a visual behavior. So I'm going to introduce now this task that we developed in which a mouse is head fixed and has a steering wheel and turns it depending on what it sees. The actual wheel was, I stole it from my son Julian's Lego box, and that's how the decision was made. He was asleep, he didn't notice, he was 10. Anyway, so the mouse is surrounded by screens. Mice have eyes on their side, and there could be a stimulus of high contrast on the left, and lower contrast on the right, or vice versa. Or there could be no stimulus on the screen. If the stimulus is on the left, is higher contrast, we ask the mouse to turn the wheel so as to bring it to the center, okay? It's a little bit similar to an orienting movement. For example, if I'm interested in something over there, I would go like this, if I'm a mouse, and my arms moved right when I was interested in something left. Anyway, but if there's nothing on the screen, we ask the mouse to not move at all. So for the experts in the room, this is a combination of a 2 alternative forced choice task and a go, no-go task. And this is crucial for our finding later, okay? So while the mouse is doing this, we can do wide field imaging of the dorsal cortex because we can use mice that express GCamp and we can point a camera to their head. This wide field imaging, by the way, has much lower resolution than 2-photon imaging, so you're not going to be able to see individual neurons here. In fact, you're not going to be able to understand much of this. It looks like very complicated, right? So I can tell you there's visual cortex down here, but it's not like you see particular activation there that stands out from the rest. The prefrontal cortex is up here. The one thing that stands out is how incredibly bilateral this activity is, right? This is the left side of the cortex. This is the dorsal side, the right side of the cortex. So let's delve in a little bit into this. Here is the average fluorescence of that mouse dorsal cortex. If we average it over 100 trials, you see the stimulus comes up at time 0. Then maybe 50, 80 milliseconds later, you see activity in visual cortex. These numbers strikes me as a little long. We have other studies in which they're a bit shorter anyway. And then you get this big activation in visual cortex, and you also see activity in prefrontal cortex, okay, which is a bit hard to see here. That's the average response. When you look at the individual trials, you see tremendous variability, okay? Here's a trial that looks a little bit like the average response, and that's the cleanest one in the entire data set, okay? Here's a trial where there's a lot of spontaneous activity frontally, and this neat activation in visual cortex. And here's a trial that is the opposite. There is this enormous bilateral activity that invests visual cortex. And we've done some quantitative assessments, and it looks like this interaction is additive. The response looks like a visual response that is like this, plus this bilateral activity. So now you may wonder, did the mouse do a better job at the task when the activity was clean like this than when it was dirty like that? Well, the answer is no. The mouse did exactly the same. So this is called the psychometric curve, how well the mouse does as a function of the contrast of the stimulus. And these two curves, this one curve comes from half of the trials where there was a lot of spontaneous bilateral activity in over visual cortex, and the other half comes from a trial like this, and the performance is indistinguishable. Some of you might be thinking, well, duh, that's because the mouse is probably not even using its cortex to do this task. And you would be wrong. I'll show you in one slide. The mouse is using its cortex, but let's first dig in and see what this activity, this visual activity, looks like. So if we go 125 milliseconds after the stimulus, we can ask, and we look at trials when there was a stimulus on the left, we can ask which pixels in cortex tell us about whether there was a stimulus or not. That's called visual encoding, and we find it mostly in primary visual cortex, higher visual areas, including some that are considered parietal, and in prefrontal cortex, although this visual activation in prefrontal cortex is only present in mice that have been trained in the task. If you were to take mice that were naive, you would only see activity in visual cortex, but of course they wouldn't be turning any wheel. So now you can ask, does the mouse actually need these brain regions? And the answer is yes. If we take a separate cohort of mice that express channel rhodopsin and inhibitory neurons, and we shine a laser on them to silence different parts of the cortex, this is a technique that was invented by the Scanziani laboratory and the Svoboda laboratory. And so we can silence different pieces of cortex and see where we have significant effects, which is the size of these dots. And for simplicity, I'm showing you the case in which we look at the effect on the responses when there were two identical stimuli on the left left and on the right. And you can see that silencing right visual cortex makes the mouse make more right choices, as if it was hard to see the stimulus and easy to see this one, okay? And you can see that the regions that are necessary are very similar to the ones that were active. In fact, there's a really nice correspondence between these data that come from mice that express calcium, a calcium indicator, and these data that come from mice that express channelrhodopsin in inhibitory neurons, and it's illustrated here. So each dot here is a little region of cortex, and the x-axis comes from this, and the y-axis comes from this. So this tells you the more a piece of cortex encodes the visual stimulus, the more we disrupt it when we use inactivation, okay? This is all very pleasing, and it looks all very logical, but things are going to start looking weird in essentially one slide, because now we're going to ask what happens when there's movement. So 125 milliseconds after the stimulus, this is the activity of the cortex. There's activity in visual cortex and in prefrontal cortex, it's all fine. And it doesn't matter whether the mouse is about to not move or it's about to move. They look similar. 150 milliseconds after the stimulus, you start to see a bit more activity maybe in prefrontal cortex if the mouse is going to move. 200 milliseconds after the stimulus, the mouse hasn't moved yet. There's a ton of activity everywhere, okay, including primary motors, somatosensory, but everywhere. In fact, no matter which piece of cortex you look at, you do a fantastic job at predicting whether the mouse is about to move or not. So this is 25 milliseconds before action. Dark means that we are really good at predicting whether the mouse will move or not, which is not a surprise to you because you can compare this to this. And when you see white here, it doesn't mean that that's a bad region for prediction. It's simply that the brain curves and we couldn't image there, okay? So summary up to here is that visual stimuli drive what the textbook says they should drive. But the upcomic movements drive the entire cortex, and that is not what a textbook will tell you. So now, given that we are in this strange territory, let's continue and ask what happens in the rest of the brain. This is about only about the dorsal cortex of the mouse, right? So we're going to now go a bit deeper in the brain by using a different method. Instead of two-photon imaging, we will use electrodes. And because we want to record from lots of regions at the same time and lots of neurons, we will use this NeuroPixels probe, which were developed by a consortium led by Tim Harris at Janelia, a consortium that includes a number of funders and institutions, including the Allen Institute, Wellcome Trust, and more. So this is a revolutionary piece of technology that is produced by actually a non-profit called IMEC, which they are leaders of electronics. And in 1 centimeter shank, they have about 1,000 recording sites, and what comes out is fully amplified and digitized. So now one of these probes can record from easily 400 neurons, and if you put 2 of them in, you record from easily more neurons than I ever recorded from my PhD thesis, which took 5 years, okay? And so this is, you can put 2 electrodes like this, and you get 800 neurons. And so Nick Steinmetz, while he was testing these electrodes, also applied these recordings to the brains of 10 mice that were performing the task that I showed you, and asked, and usually he would do one NeuroPixels probe at a time, or 2 at a time, but never more than 2, I think. And this gives you an idea of what regions he covered. They were all in the left hemisphere. He stayed out of cerebellum, which we now know actually is super interesting in this task. And they don't cover the entire brain, but he did his best. He was one postdoc, right? And so now I'm going to summarize what he found by doing a little bit of what the fMRI folks would do, which is to show you maps of brains and tell you where there was activity and where there was not activity, okay? But remember, this is activity at the level of individual neurons. So you will see some maps where I say 2% of the neurons responded, and half of them did one thing and half did the other. That is something that you would never be able to get from fMRI or the equivalent in mouse, okay? So first of all, let's ask, where did Nick find visual encoding? Reassuringly, he found it mostly in visual cortex, prefrontal cortex, you've already heard this, superior colliculus, which is down here, it makes a lot of sense. He also found it in some midbrain regions, which, you know, it's fine. It will look a bit surprising to you, maybe, that the whole striatum seems to respond, but we actually made no effort here to parcellate the striatum more finely, and I bet that there was only one small region of the striatum that responded to vision. So anyway, this makes a lot of sense. The winner was visual cortex, although it had a bit of an unfair advantage because we aimed the electrodes in the place that would look at the stimulus. And this looks extremely similar up here to the data I showed you before with the wide field imaging, right? You see these agree very well. Now let's ask, where did Nick find action encoding? And by this I mean neurons that fire if the mouse is about to move the wheel, regardless of whether the movement is to the left or to the right. The answer is everywhere, in the entire brain. There's not one region where Nick recorded where he didn't find neurons that are about to tell you, they're telling you if the mouse is about to move or not. And the winner is the anterior pretectal nucleus. And again, I bet there are posters at this meeting about the anterior pretectal nucleus. That's where we found the most neurons that encode upcoming movements. So again, this is, when we look at cortex, this is very much in agreement with our wide field imaging, right? There's not a single piece of cortex that is not good at telling you this. And I think, if I may just make a little philosophical remark, that this finding is the solution to what is otherwise a great mystery in neuroscience. And the great mystery is that everybody who has recorded from any brain region during any behavior has been able to write a paper. And that's my feeling for why that happens. So now you may be curious, but what neurons make the choice about moving left or right? Choice is not the subject of this talk, but I will show you. They are really rare, and they tend to be in the same regions where we found visual responses. The strongest activity is in the midbrain, and the winner is zona incerta, a region that I didn't even know before we did the study, where 6% of the neurons tell you if the mouse is about to turn left or right. And the neurons in prefrontal areas are more rare. And finally, there's something that is quite topical, because I think that I've probably lost a third of you, to be generous. And so a third of you are disengaged, let's say, and 2/3 of you are engaged. If I looked at your brain right now, what difference would I see? So if you are mice, or I think if we agree that the mammalian brain follows the same plan, this is probably what the brain looks like of those of you who are engaged versus disengaged. So blue are regions that fires more when the animal is in a passive state, so not doing the task at all, and red are the regions where the neurons fire more when the animal is in the task. And you find that cortex generally fires less, which is something that Tony Zader had observed, and people yelled at him a few years ago, but he was right. And instead, some subcortical regions, including the dentate gyrus and the striatum and other regions, fire more when the animal is in the task. And I'm talking about firing before a visual stimulus shows up. Okay, so this is not about movements or stimuli. This is about tonic firing. Let's summarize. I showed you that signals related to brain state and upcoming actions are present throughout the brain. I also showed you that these bilateral activations don't interfere with visual behavior. And so now you may wonder, might they interfere with our job as neuroscientists? In other words, if there's all this activity related to upcoming movement, and we're not measuring the movements of an animal, might we get confused? And the answer is yes, we can get super confused, okay? So here is an example of a study where, I hope he won't be mad if I say, Célian Bimbad, fantastic postdoc, he came to my lab to study this phenomenon that has been known, which is that if you present sounds, you get activity in visual cortex, okay? Which has been taken to indicate that the cortex is extremely multisensory. And this is a finding that we replicated. So Célian presented these different sounds and recorded this activity in primary visual cortex. It was extremely stereotyped across neurons and across mice. So we then wonder, but could this be that these sounds actually evoke small movements? And I'm talking about tiny movements, okay? So if you look at the face of the mouse, it's got these whiskers. It turns out that these sounds are accompanied by little twitches of the whiskers. And these twitches, if you measure their energy, are extremely similar to these responses that look like responses to sounds. Now, clearly, it's the mouse brain that is making the whiskers twitch, okay? Don't get me wrong. Those are our auditory responses. But I would say that given the talk you've just heard, it is a more, what's the word, parsimonious explanation to think that it's not the sounds that drive visual cortex, but rather the sounds evoke tiny brain body movements and changes in brain state, which in turn affect visual cortex. And in this paper, we actually test this by cutting the connections from auditory to visual cortex, by recording in lots of places and doing various other controls. So finally, I would just want to show you a couple more slides that go in the question of what kind of signals are broadcast. Because up to now, we've only looked at signals related to brain state, arousal versus drowsiness, and upcoming actions, turning a wheel versus not turning it, or twitching your whiskers. And I've told you that these are present throughout the brain and make our job as neuroscientists much harder. Because it's hard to distinguish what's due to movement and what's not. But then the question is, are any other signals broadcast so widely across the brain? And so for this last couple of slides, I want to show you results that we obtained by putting mice in virtual reality. And so we have mice running on a running wheel, and they are in a virtual corridor. And the corridor has first some bars that you can see here, then a plaid, and then it repeats, bars and a plaid. I think it will show, yeah. Okay, so plaid, bars, plaid. Well, sorry, something of that sort. I think I only have half of it. Oh, no. Yeah, I only have half of it, sorry. Short movie. Anyway, trust me, the corridor is visually identical for 40 centimeters, and then again for another 40 centimeters. So the prediction is that if a neuron in visual cortex were purely visual, it should respond identically in the first half of the corridor and in the second half of the corridor. And this is true for some neurons. However, we also found various neurons that fire more in one half of the corridor or more in the other half. And we looked carefully about whether this might have to do with running speed and things like this, and none of that explained it. So if we now look, instead of these example neurons, we look at a whole population, we find that primary visual cortex is tiles the space that the animal is running through. Some neurons prefer early positions, some neurons prefer late positions, and the responses do repeat themselves a little bit in the first half or the second half. But some neurons clearly fire more in the first half rather than the second, and some neurons clearly fire more in the second half rather than the first. And as a control, or a scientific question, we asked, would it be the same in the thalamus that provides input to visual cortex? And the answer is no. We imaged the spines, the boutons sent from lateral genicular nucleus to visual cortex, and we found that those boutons responded equally in the two halves, okay? All of this is quantified in these studies, by the way. I'm just showing you the data qualitatively. Moreover, if we go beyond primary visual cortex to these other areas, we find an effect that is very similar to the one present in primary visual cortex. It's almost fully formed there. And these results are now adding to a growing body of data indicating that navigational signals are shared widely across the brain. For example, Cindy Poo in the Mainen lab recently published a paper about how the olfactory cortex has signals about where the mouse is, navigational signals. And so it seems like these are broadcast very widely. So here is a summary of the whole talk. And so we looked at the fact that visual signals, and you will be pleased to hear and to remember that some of these signals are nicely localized, like the textbook says. For example, visual signals are mostly in visual areas. that's fine. But other signals, surprisingly, are broadcast extraordinarily widely across the brain. For example, signals about brain state and impending actions, and even about navigation, which, textbooks tell us that it's the province of the hippocampus and entorhinal cortex, etc. I showed you that at least in cortex, these visual signals and non-visual signals interact through addition and multiplication, and probably they do this through the action of diverse inhibitory neurons, which can be organized on a single axis to describe their actions. And so I want to tell you that overall, one of the lessons that I learned from this is that studying large populations across the brain is great fun, but it is also fun, especially when we join forces across scientists. Now, we all join forces anyway, because in a single lab, the PI joins forces with postdocs and students. I personally have found it super useful to join forces with Kenneth, and I think I wouldn't have done a tenth of what I showed you today if we hadn't shared the lab. And moreover, I think we can go further and join forces on a broader scale. And I've done that by joining the International Brain Laboratory, which is a cooperative, if you want, of 22 labs, half of them theoretical and half of them experimental. And the IBL has just released an incredible data set on a task that is a bit more complex than this one. It has a cognitive component. And I invite you to visit the IBL at booth 3330, because also if you want to talk to me, that's where I'm going to be after this talk. And so I invite you to visit it as well.